Hello everyone, myself Hemant Kumar, student of MTech Dairy Technology. I am in second semester. My topic is safety indicator organism Bacillus cereus. First one, introduction. Bacillus cereus is a foodborne pathogen that can produce toxin causing two types of gastrointestinal illness. The first one is emetic syndrome and the second one is diarrheal syndrome. When the emetic toxin is produced in the food, vomiting occurs after ingestion of contaminated food. The diarrheal syndrome occurs when enterotoxins are produced in the intestine following ingestion of food contaminated of Bacillus cereus. Food poisoning caused by Bacillus cereus is an acute intoxication that occurs when these microorganisms produce toxin causing two types of gastrointestinal illness. The first one, emetic syndrome and diarrheal syndrome. In Canada, over 36,000 cases of foodborne gastroenteritis worldwide. In Bacillus cereus were estimated to have occurred in 2006. Bacillus cereus will grow in food that has been improperly stored. Therefore, proper food handling, especially after cooking, will help prevent illness caused by this microorganism. This is the picture of Bacillus cereus. This is shown by microscopic picture. Symptoms include in emetic syndrome, there is vomiting, nausea, sometimes diarrhea. Symptoms usually start 0.5 to 5 hours after ingestion of contaminated food. Usually, symptoms disappear in 6 to 24 hours. Diarrheal syndrome. In syndrome, diarrhea sometimes is blood and or mucus, nausea, and may also include abdominal pain. Symptoms usually start from 8 to 16 hours after ingestion of contaminated food. Usually, symptoms disappear in 12 to 24 hours. In some cases, the illness may be more severe. If you have serious symptoms, you should see your doctor. Causage. Bacillus cereus is widespread in environment and commonly found in the soil. It is able to produce spores that are resistant to heat and desiccation. Therefore, it is not uncommon to isolate it from both raw and cooked food. These spores will germinate into vegetative form of Bacillus cereus and grow if the food is held under favorable condition of pH and temperature around 8 degrees Celsius and 55 degrees Celsius for a sufficient time. Although the presence of vegetative form of Bacillus cereus in food is always necessary for foodborne disease to occur, not all the Bacillus cereus strain can produce the toxin that causes the emetic of diarrheal syndrome. The diarrheal syndrome occurs when a large number of vegetative cells of Bacillus cereus are ingested and produce enterotoxin in the small intestine. A wider range of food have been linked with diarrheal syndrome, such as meat, stew, soups, sauces, vegetable and milk products. Complication. Both the emetic and diarrheal syndrome are usually self-limiting, resolving within 1 or 2 days. In a small percentage of cases, it can be more severe, and deaths have been reported in the literature. The elderly and patients with lower stomach acidity may be more susceptible to diarrheal syndrome. Test and diagnosis. Food poisoning caused by Bacillus cereus can be confirmed by isolation of this bacterium in food stool or vomit sample. Different media are available to culture this pathogen that commercial kits are also available to detect the diarrheal enterotoxin, but this is not the case for emetic toxin. Treatment and drugs. Most people recover without treatment. The administration of fluids is recommended if the diarrheal or vomiting is harsh. Antibiotics are indicated as the symptom are caused by toxin and not the bacteria. Prevention. As Bacillus cereus is ubiquitous in environment, control measures should be focused on preventing growth of Bacillus cereus and the formation of emetic toxin in the food. Ensure food is maintained either at a temperature above 60 degrees Celsius or refrigerated below 4 degrees Celsius. Full cooked food that will not be immediately consumed to below 4 degrees Celsius within 6 hours. When reheating food, ensure that the temperature reaches at least 74 degrees Celsius. Examination of food for Bacillus cereus. Sampling. If the quantity of food to be examined is large, take representative sample of 50 gram each different part of the suspect food because contamination may be unevenly distributed. If the food is powder or consists of a small disaccharic particle, then it should be thoroughly mixed before taking samples. Transporting and storage of samples. Transport sample promptly in insulated shipping container with enough gel type refrigerant to maintain them at 6 degrees Celsius or below. Upon receipt in the laboratories, store the sample at 4 degrees Celsius and analyze it as soon as possible. If analysis cannot be started within 4 days after collection, freeze sample promptly and store store at minus 20 degrees Celsius until examined. Thaw at room temperature and proceed with analysis as usual. Maintain frozen samples at 20 degrees Celsius until examined. Sip on dry ice to avoid thawing. Dehydrated food may be stored at raw temperature and sipped without refrigeration. Detection of Bacillus cereus in traditional method. In traditional method for Bacillus cereus detection is agar plated based counting for which the guidelines are provided by the ISO 7932-2004 that had been last revised nearly 15 years ago. Several laborious points are needed like sample homogenization, numerous configuration and serial dilution preparation all carried out in aseptic condition to avoid any contamination. The plating steps have to be carried out in duplicate for each dilution using specific media. Then plates need to be incubated at 30 degrees Celsius for 24 hours to 48 hours. Finally, colony have been streaked out on the brain heart infusion agar medium. Moreover, an additional confirmation assay is required in order to differentiate bacteria of the Bacillus cereus group. This last step takes another 2 hours to 24 hours. Traditional methods are thus time consuming, laborious, expensive, and require trained staff capable of ensuring the correct procedure. Furthermore, with these methodologies, it is not possible to detect injured or behavioral but not culturable cells. The ISO 7932-2004 method is specialized a horizontal test for immunization of presumptive bacillus cells. This method consists of a serial dilution of the samples and a subsequent dilution spreading on bacillus cereus selective. Conclusion: Bacillus cereus is an important cause of foodborne pathogenesis because it contaminates numerous food including infant formula, infant rice cereal, milk and dried milk products, rice, egg, meats in pathogens. Bacillus cereus strains causes food poisoning or even human infectious disease with an infective dose as low as 10 to the power 3 CFU per gram of food. Although Bacillus cereus is currently searched only in infant formula, it is recommended as a suitable microbiological safety indicator for food products. The traditional detection method that enumerate Bacillus cereus colony on selective agars are labor, cost intensive, and time consuming. Furthermore, they cannot detect injured or BB and C cells. Thank you, everyone. Thank you, sir, my batchmates. Hope you understand about safety organism Bacillus cereus.